I am just getting everything set up here. So today we are going to continue our talk that we started on Monday. So just kind of addressing the muddiest points, but we actually got through a lot of the muddiest points that we had identified from last week. And so today I'm pretty excited about today because a lot of the stuff that I have identified to talk about today is sort of just like fun stuff, sort of the instead of strictly the questions out of confusion, which are always very important. Today, I think we get a little time to talk about the questions out of curiosity, which I love, because there were some really good ones. But before I get into that, are there any questions about this week, or I can kind of show you what we covered on Monday if there's any additional questions. Let's see, about anaerobic cellular respiration or trophic levels or photosynthesis, anything that we talked about on Monday before we get into just, again, like sort of the fun stuff. Or I can also mention this week, obviously, we are talking about transcription and translation. Next week, we will be continuing transcriptional and translation, except for we're going to talk about how it's regulated. So Professor Gillen Wiseman kind of took the beginning of that this week, talking about the processes of transcription and translation. And then I'm excited because I get to take over next week and talk about how those processes are regulated, which I think really sort of like solidifies those processes and what they mean and why they're so important, which is really where I'm passionate. So really good stuff. But are there any questions right now about this week or last week or anything? Everybody feeling okay? All right. I do have some very exciting news to share. I am so excited about this. This was announced in via ASU Now email today, and I'm like, I'm having trouble concentrating because I'm so excited. You all are very well aware how much I love Crash Course and Hank Green of the Green Brothers for his wonderful Crash Course in Biology videos, stuff like that. Well, imagine my surprise when I woke up this morning and I saw this article in ASU Now saying that the Green Brothers and ASU are partnering to create high quality videos. It's called ASU Study Hall. And I just want you to notice that right now there's one starting for chemistry, data literacy, composition, and algebra. And I'm a little freaked out because there's not one for biology. So I have to find out if there will be one for biology, because if there's going to be one for biology, it has to be me. Like, I cannot handle this. I'm freaking out about it. I'm so excited, although, like you all know, that might be bad news for my husband, because I've told you his only competition is Hank Green, this guy right here. I sent him an email this morning and he was like, oh no. As much as he could say. He is, as several of you know, in medical school right now. He's doing his rotation in pulmonary critical care, so working with COVID patients. So I think that's the only thing he could say right now. Oh no. Like, that's right, watch out. But I am so excited about this. I think this will be wonderful. They probably already have somebody identified for biology, but I don't know. If you know who to talk to about this, Sean Hobson, Chief Design Officer for Ed Plus, send them my name and tell them how enthusiastic I am, but don't tell them about my giant embarrassing crush on Hank Green. Maybe leave that out. But I'll pop that in the chat just if you want to see that is very exciting news. I think it'll be really wonderful. I mean, that basically means that ASU is going to have their own crash course, which is beyond amazing. So anyway, I had to share that news with somebody. Yes, Abby, yes. Please. Thank you. I am on board, you know. Okay. So I had a question in the chat. So what is crash course? Let me show you. Let me put a link here. I'm gonna change your life. Crash course in biology is a really wonderful source for tools for biology, including this course. I mean, I think it's one of the best ways, kind of supplemental biology knowledge. Hank Green, and the production quality is awesome. Hank Green is just an incredible scientific educator, scientific communicator. And you can go to these pages, and if you go to Crash Course, they have all of these different, oh look, study hall's on there. I'm so happy. But you can see they have tons of videos on just random things. John Green, who is an author as well, also does a lot of like social sciences crash course videos. And Hank Green does a lot of the science side of videos. So I obviously like have a really, I'm familiar with him because of Crash Course in Biology specifically. And if you go to playlists, let's see if you can find it. If you go to playlists, well, they have like a million playlists, but they have Crash Course in Biology. I mean, here's a really good example of one, which is, gotta wait for a second to get through this, but here's a really good example of one. That's Hank Green. And I'm just kidding. But here's a really good example of one, which is so good. It's ATP and respiration. So he talks about it at a level that I think is excellent for this class. So I try to push out the videos. Amoeba Sisters are another really good one, but Crash Course is my favorite. Sometimes, much like you might have to do to my videos, it is nice to have the option to like slow it down, like go playback speed like lower, because he talks really fast. But aside from that, his videos are phenomenal. So I really recommend checking that out. I'm like so excited about this. Too excited about it. Embarrassingly excited about it. So yeah. Let's see. Yes, agreed. He is smart and cute. And you know, like, yeah, I agree. Like, smart is cute. So both. So yeah, so I know, I'm so excited, you guys. Embarrassingly excited. So check that out. I posted the link in there. And yeah, and I mean, I'll post a link to this crash course for ATP and respiration too, because that's a really good one. So, but yeah, you'll notice at the end of all of my Muddiest Point lectures, let's see if I have it pulled up, but you'll notice at the end of all of my Muddiest Point lectures, I'll always have, and several of you post it too, but you'll always see me post a crash course reference video. They're so good, and they have them for every subject too. So I love it. I love it. Okay. Now that I got my opportunity to fan out, or yeah, and now I can like try to breathe. Let's talk about some more fun stuff. I mean, it's a fun Wednesday. I'm really happy. All right, so again, if you have more questions sort of about the Muddiest Points, We can totally talk about it, anything that's going on in the class. But if not, you know, let's talk about some really fun stuff. Let's talk about the questions we got out of curiosity. So we did have a really good question by Jared, which was, why are the light and dark reactions physically separated? So remember that the light reactions take place in the thylakoid membrane, whereas the dark reactions take place in the stroma, so within a chloroplast in eukaryotic cells. So why do they take place in different locations? And I really loved this question because it means that I have the opportunity to talk about the importance of structure versus function. So that'll be really fun. And then also a really good question by Heather was, how long can an organism perform anaerobic cellular respiration before dying? I love that question as well. Really interesting question. Also allows me to talk about bacteria. So love that. And then I also had some really good questions, actually from several of you from a couple weeks ago. And then my other class asked this question a lot as well. So I thought it would be a really interesting thing to discuss, which is, could we potentially genetically engineer animals like ourselves to contain pigment like chloroplasts, and allowing us to perform photosynthesis, generate our own food? And so I thought we could talk about that. I wanted to talk about one other thing that was a pretty interesting conversation that I had with somebody yesterday. And I think this is a good kind of pickup to our next question. But somebody asked me yesterday, or when I was explaining the Calvin cycle yesterday, and as you all know, we found out in Bio 181, sort of contradictory to a lot of the, you know, kind of like general explanations of photosynthesis that we get throughout our lives. This is the first time that, I know that the first time I found this out was actually in graduate school, which is that the process of the Calvin cycle, and therefore the process of photosynthesis, doesn't actually generate glucose directly. And instead what happens is it takes carbon dioxide, fixes it to an organic molecule. So this is like arguably the most important process for photosynthesis, aside from like being able to use light energy instead of chemical energy. But with photosynthesis, we can take gas carbon source, so CO2, we can take that gas carbon source, and it is via carbon fixation, which again only occurs in the Calvin cycle. So not only in plants, because remember, other organisms can do the Calvin cycle, like prokaryotes, like phytoplankton are one of the like most prevalent organisms on the planet that do photosynthesis and therefore carbon fixation. But because of autotrophs, which do carbon fixation, we can take gas carbon sources, CO2, and turn them into like liquid or solid state, soluble forms of carbon. So that's what we talk about when we go from like inorganic carbon to organic carbon. That the reason that's sort of like an amazing thing is because actually we're going from like gas carbon source to a soluble like liquid or solid form, which not all organisms can do. Like obviously, if you think about it, like if this was an easy process, this would mean we could like breathe in carbon dioxide and use that for biosynthesis. We obviously cannot. Carbon dioxide you should not breathe it. It is toxic to breathe in high quantities, right? You need oxygen. But this is what essentially a lot of people, you know, up until this class, I think the majority of people see photosynthesis as its primary purpose is to generate glucose or to generate sugar. And that's not even what it does at all. Photosynthesis as a process, its main purpose in the ecosystem is not to generate sugars. It does not directly generate sugars. Its main purpose is to take gas carbon and turn it into a soluble form of carbon. So to perform carbon fixation. So again, glucose is not even directly generated. It actually generates glyceraldehyde 3-phosphate, so G3P, which can be further utilized to create sugar, right? So two glucose 3-phosphates can come together to form a glucose. That's a separate process that occurs like after photosynthesis. But all that photosynthesis does, I guess, when there's a Calvin cycle, is it does take gas carbon sources and turn it into a soluble form. So overall, through the dark cycle, we actually get 12 glyceraldehyde 3-phosphates. Actually, only 2 of those exit the cycle for the creation of sugars. 10 of those actually re-enter the cycle to allow the cycle to continue with its primary purpose, which again is to turn gas carbon sources or inorganic gas carbon sources into organic soluble carbon sources. So the reason I wanted to bring this up is because I wanted to reiterate that kind of one of those, what I think for me as a student was one of the most revolutionary things to kind of like, or I sort of like mind-boggling things to learn about photosynthesis, which is that the primary purpose of plants is not to be eaten. Plants do not exist for our consumption. We typically think of them, right? We think about the fact that plants take carbon dioxide and turn it into glucose, and then we can eat that glucose and use it for our purposes. But in all reality, like in an ecosystem, the purpose of plants is not strictly to like support, you know, like our energy, you know, needs. And in fact, its purpose is to create, you know, very importantly, to turn gas into soluble carbon. So I mentioned this and somebody was like, well, this brings me to a question that I've had for a long time, which is this, you know, like knowing that plants are not like their sole purpose is not necessarily for our consumption. That makes a little more sense, right? If you think about like onions, what happens when you cut an onion? Did you like cry? It smells terrible, right? A lot of people like raw onion. I'm not one of those people. I wish that I did, but like, I can't stand the taste of raw onion. I cry, I sob when I cut onions. Like it's so bad. I can't, my eyes are so sensitive to it. But that is literally a defense response of an onion. It is a chemical defense to try to dissuade that onion being eaten. Same thing with like hot peppers, capsaicin. That's a defense mechanism to like prevent it from being eaten. I also, like, for example, grass. I watched this really cool video a really long time ago, as you all know that I really love plant communication. And it turns out that being from the Midwest, we have lots and lots of grass. And it's basically all we have is like just tons of grass, maybe some cows too. No, just kidding. I love the Midwest. We have some stores, but we have a lot of grass. And so one of my favorite smells is like when you mow the grass, the smell of freshly cut grass. Like it smells so good. And I watched this video about plant communication, and it turns out that that smell is actually a chemical produced by the grass to warn surrounding plants of danger. So the smell of fresh cut grass is actually like grass screaming for help. I can't laugh. Like that is so sad, I guess, for the grass, but that was sort of one of those things where it's like, wow, like, you know, these plants, you know, exist outside of, you know, just making like yards pretty. So I will say that like, we do like consumption of plants by animals, Oh, Carly, I didn't know the only way to get nutrients from spinach is to cook it. I had no idea. Wow. They protect their nutrients. That makes sense unless we cook it. But yeah, there are some plants where consumption of those plants helps to like spread their seeds. Um, but for the most part, like the fruit of the plant, which is what we eat, is to kind of like help spread their seeds. So you get a really heavy fruit, it drops the seeds into the soil and it perpetuates the cycle of life. But anyway, I just thought that was kind of an interesting comment, that when we talk about photosynthesis and we talk about plants and how they can create glyceraldehyde 3-phosphate, they don't directly produce the sugars that we eat, but their main purpose is carbon fixation. I thought the comment that onions and peppers are a little bit difficult to eat was a really important, like, interesting thing to support that. So I thought I would share. Interesting conversation.
I had a quick question.
Yeah.
Since like through the process of like climate change with carbon emissions being like the main cause for it, is there any way we can artificially mimic like that Calvin cycle to change the states of carbon?
That is such a good question. And the answer is, yeah, we're actually trying. So I'm going to share my screen, and I'm just going to do a Google actually, because we are actually trying to use like these giant tanks full of algae. Let's see if I can find it. Yeah. So this is an example of that. So this is just like an algae field, and all of these are individual tubes trying to sort of like maximize exposure of the algae to the sun. And the whole purpose is to hopefully actually derive, because if you remember from the beginning of that metabolism lecture, like gasoline and well, actually glucose, but the products of reactions beyond photosynthesis are actually very similar. And so they could potentially be used to like fuel like cars in place of gasoline. So we're trying to extract the products here in these like in these giant like out biofuel containers. But another really big thing that we want to do with these big algae fields is exactly that, like try really hard to reduce the carbon dioxide in the atmosphere due to pollution. One of the big problems with global warming, and especially one of the issues that's been ongoing for many years, has been deforestation. So rainforests were one of the largest areas for photosynthesis, and so we're one of the biggest contributors to sort of like reducing CO2 and therefore reducing the warming due to CO2. But now so many like deforestation has devastated rainforests and forests across the globe actually, and so I think that, you know, that's one of the hopes with these algae fields. I have a question. I mean, raise your hand if you have ever seen or heard of those algae fields or biofuels. Raise your hand electronically. I'm not seeing any hands. You know, that's oh good. Okay, I'm seeing some hands. That makes me happy. They
Just to comment.
are, oh yeah.
Sorry, just to comment on what you said. I think the biofuel using algae for the biofuel, it was actually in the Labster, one of the Labster labs.
Oh, cool.
So
Good.
yeah, I just wanted to comment on that.
Thank you. Yeah, that makes me really happy that it is in one of those Labsters. Good. But yeah, it's the frustrating thing about this is that, Ren, you're right, like this could be like an amazing, not only biofuel, but also way to kind of reduce global warming, but they are not very prevalent. So that's one of the reasons, you know, to not to get political, but to invest in biofuels and green energy. That might be a really important thing. So hopefully investing in those would make them more prevalent. Basically.
Yeah, I just had that question because I know that we're trying to like artificially create other things organically, but I was wondering like if we could mimic the photosynthesis process and like create our own Calvin cycle, you know, stuff like that.
Oh, so do you mean like in us or just like in nature?
Just like, I don't know, like anything, like create like a product like that changes the states or something. I don't know.
Oh, yes. I think that we can. It's just challenging, and I guess I will say that I don't know enough about. I think that's like the purpose of like bioreactors and tools like that. Like I do know that it is possible, but it's just not as efficient as just like using like algae or trees, I guess I should say. So it's a really good question. Yeah. All right. I love those questions. Good job. All right. So let's look back at our slides. So cool. So I wanted to visit that. And then again, we had a question from Jared that was kind of following up on that. So again, we do know that photosynthesis occurs in two reactions, the light reactions and the dark reactions. The light reactions are those photosystems which generate ATP and NADPH, and those occur in thylakoids. And the dark reactions occur in the Calvin cycle, which is in the stroma. So these are in different locations in the chloroplast. So the question was, why? And the answer is a really cool one. And I love that this question was asked because it doesn't get asked a lot, but when it does, I love talking about it. Because I want to remind you that, so what we are looking at here is the light reactions. And this is in the thylakoid. And remember, the light reactions, photosystem 2 specifically, culminates in the production of a proton motive force. So what is happening here is light is absorbed, it excites electrons, and electrons pump hydrogen ions, which remember, hydrogen ions are essentially protons because they're an ion, meaning they've lost an electron, which is why they have a positive charge. Hydrogen ions also don't have a neutron. And so, well, I guess hydrogen in general doesn't have a neutron. So what happens with hydrogen, if it loses an electron, all that remains is one proton. So when we talk about a proton motive force, it's literally just like pushing hydrogen ions to one side of a membrane. But because those hydrogen ions are literally just protons, we call it a proton motive force. So a proton motive force is essentially just like a concentration gradient of hydrogen ions. So that's what you're seeing here. And just like we saw with the electron transport chain in cellular respiration, this is an electron transport chain that culminates in a proton motive force that will basically fuel the production of ATP from ADP and phosphate. So you see this is like that proton motive force, the Hoover Dam of hydrogen ions, fueling the phosphorylation of ATP. So one thing I want to remind you about those hydrogen ions is not only are they useful for a proton motive force, but if you think back to like way earlier in the semester, hydrogen ions are also in combination with hydroxide ions used to determine pH. So if you remember, our pH scale, so whether something has a low pH and is acidic, or whether something has a high pH and is basic, or whether something is neutral, all depends on the quantity of those hydrogen ions. So the question was, why would the why are the light reactions and the dark reactions separated? One of the answers has a lot to do with the fact, sorry, I thought I had that off. One of the reasons has a lot to do with the fact that these chemical reactions require like a pH that's relatively neutral. Like the Calvin cycle has to take place in a neutral environment. So the stroma is a neutral pH, about a pH of 7. But because of the fact that in the thylakoids we're generating this proton motive force and having all of these hydrogen ions present, what happens in the thylakoid lumen is the presence of those hydrogen ions actually makes this thylakoid lumen slightly acidic. So the lumen can handle that, right? It's meant for that. So structurally, it's meant to tolerate, and also the reactions that take place in the lumen can tolerate that low pH. However, the Calvin cycle could not take place in that acidic environment. So we have to have separation of these reactions. As well as that, the physical separation of these hydrogen ions is not only important for establishing that proton motive force, but also for containing the acidic environment that's established in that thylakoid. So making sure that the stroma of the chloroplast is neutral, whereas that acidic environment is kind of contained in that area. So structure, that's like, I go back to structure equals function. You will need the membrane of the thylakoid to generate a proton motive force. This also does result in sort of like an acidic microenvironment, which needs to be contained so that the other reactions are not affected by that acidic environment. So one of the questions I get a lot with this is like, if that's true for the chloroplast, is that also true for the mitochondria? And the answer is yeah. So the interior of mitochondria are slightly acidic because of the fact that you're generating proton motive forces across those membranes. So I saw a lot of questions. I'm going to pop them open. So let's see. Oh. If there is a fire alarm going off, I'm so sorry. Yes, you need to exit the building. This is recorded, so don't worry about that at all. Don't ever worry about that. But yes, you need to go outside. There should be a parking lot identified. Please send me an email, Jordan, to make sure everything is okay. Okay. So Sophia, you asked, is it basic in, oh, no, acidic. Yes, exactly. It is basic. That means that, yeah, inside of the chloroplast, this would be an acidic environment. Yeah, because of all the hydrogen ions. So that is, again, like why it is so important to remember that like structure equals function. This is one really good example of that. Let's see. And the same for the mitochondria. Sorry, I got distracted. Okay, I hope everybody is okay. Another question, which was Heather's question, was how long, so we do know that if an organism doesn't have oxygen, how long can that organism survive performing anaerobic metabolism? So either anaerobic cellular respiration or fermentation. And the answer is, again, structure equals function. So this completely depends on what tissue is having a lack of oxygen. So certain tissues are less energetic than others, so require less oxygen to produce less energy than others. So for example, like nervous tissue, because action potentials require so much ATP, right? It's like charging a battery all the time, right? So nervous tissue, a lack of oxygen, nervous tissue will start to die in around 3 to 6 minutes in a lack of oxygen. They cannot perform anaerobic or really fermentation for very long at all. The kidneys and the liver are also quite energetic, but they can survive about 15 to 20 minutes without oxygen. So a lack of oxygen is called hypoxia. Heart tissue is made of cardiac muscle, and cardiac muscle in general requires quite a bit of energy, but muscle is sort of characterized by being able to supplement with anaerobic respiration and fermentation. However, not for very long, because it does require a significant amount of ATP, and the accumulation of lactic acid will eventually kind of lead to an acidic microenvironment, which would start to denature tissues. So heart tissue can last about 30 minutes in a hypoxic environment, not very long. Skeletal muscle, though, which is the muscles associated with your bones for movement, has the possibility, because of the fact that it kind of depends on what you're doing, but 60 to 90 minutes of hypoxia, which is why, you can have endurance athletes doing pretty extreme, stuff for quite a bit of extended time. Smooth muscle or vascular muscle, which is what is utilized for blood vessels and stuff like that, that can actually last quite a while without oxygen. And that's because like vascular muscle and smooth muscle don't actually contract really. The smooth muscle is not contractile. So it is pretty much just sort of very similar to like epithelial, just a little bit stronger. And so this can actually last 24 to 72 hours without oxygen. So it does completely depend on tissue. And the key here, and we're going to talk about genetic regulation next week, which I am really excited about, but the key is that what happens with your body. So for example, if you get into a situation where oxygen is in a lower concentration, so let's say that you go to like Colorado and you go hiking and there's just like less, like if you go to a higher elevation, it's like less oxygen. And if you do that, a lot of people get altitude sickness, and it's because there's like less oxygen, you're having trouble getting oxygen to your tissues. Characteristically, one of the biggest signs of that is just like extreme fatigue. And that tiredness is actually your body trying to, what it says, down-regulate your metabolic rate. Like literally your body is trying to like slow down your metabolism so that you require less oxygen. Your body will upregulate like the production of hemoglobin. You'll produce more red blood cells. You'll try to produce more things to carry more oxygen through your body, but it will just take that takes more time than it does to just say like, let's slow down, right? So I guess short answer, like without oxygen, a human will not survive very long. But what about prokaryotic organisms? They can survive a lot longer. For example, there are prokaryotic organisms that have survived. We were able to find them in these like salt crystals that when we did carbon dating, we found that these bacteria were about 250 million years old. And we were able to pop them in a broth and they came back to life. As soon as we gave them oxygen, they were like, oh, great, like things are good again, here we are. And so prokaryotes can obviously survive a really, 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 like millions of years without oxygen. So how do they do that? Just like us, they would slow down their metabolic rate, so how much ATP they need, but they also produce something called an endospore. So this is actually the endospore inside of a bacterial cell. Not all bacteria can do this. Clostridium and Bacillus. So some examples of these I'll talk about in a minute. I think I have time. Yes, I have time. But just to kind of give you an idea about what an endospore is, you don't need to know any of this. I'm just telling you for interest. what a cell does in order to survive, what a bacterial cell can do in order to survive without oxygen for a long time, is the cell basically will create a separate, like, new strand of DNA. It will like start surrounding that DNA with like a protective, really waxy, water resistant coat. And then it will actually just like only exist as that waxy little coat structure. And this is called an endospore. So these bacterial cells have such, they're first off, they're like coated in like the thickest, waxy, like environmentally resistant coating possible. But on top of that, they expel a lot of water. And so it's a really kind of like a dry environment inside of the cells. So these cells are not, or these endospores, I should say, are not really prone to any damage as far as like hypertonic or hypotonic solutions or different salinities, which is why those endospores mentioned here survived in like salt crystals. That's like the most hypertonic environment you can get, but they didn't dry out because of this coat, which and less water preventing the preventing any hypertonic or hypotonic changes. And then as well as that, they basically just turn off their metabolism. So they have some ATP in there, but they're not doing protein synthesis. They're not creating new proteins. They're really not doing anything. Like they basically just like turn their metabolism off. They're just like sitting there, not doing anything, protecting their DNA. And what can happen is the only thing that they will have on essentially is a gene that basically allows them to detect whether it's like favorable conditions. And if this cell gets in favorable conditions, then it can just like come back to life, essentially. So like I said, not all bacteria can do this. Archaea are pretty good at doing this because they can survive pretty extreme environments. But they're, as you can imagine, if a bacteria is able to form an endospore not only protects it from like drying out, an endospore also protects it from like antibiotics and bleach or any kind of thing that you can think about trying to eliminate it. So just a warning, the bacteria that can do this tend to not be the most ideal for infection. So just a warning again, if you have trouble with like graphic images, I'm going to show the next two images are a little bit graphic. But if you're interested in that, that's okay. Hang on. So one of the sort of notorious bacteria that can create endospores is actually called Clostridium perfringens. And this is the organism that causes gangrene. And so what happens is this bacteria gets into the bloodstream, or if it gets into the bloodstream, is it actually cuts off blood supply and therefore oxygen to the tissues. So this is actually what tissues will look like if they don't have vascular or blood supply. So you can see the black coloration, that is tissue death because of the fact that they're not getting nutrients via blood supply to that tissue. So gangrene is actually pretty rare. It was a lot more common in the past where like shoes and protective gear, it was commonly associated with like wartime wounds whenever people would have exposed wounds to like soil. Because Clostridia, these bacteria are very prevalent in the environment, but they have to be like, if they enter an organism, it's through like a puncture wound. So these organisms actually cannot survive in the presence of oxygen. So if the question is like, why would this organism get into like a wound and cut off oxygen supply to that tissue, it's because oxygen actually kills Clostridium perfringens. And so it cuts off blood supply to this tissue because it's trying to eliminate oxygen getting here so that it can survive. So that's gas gangrene. Pretty rough stuff. Pretty not very common. At this point, gangrene would definitely be caught before this time point. This is a very extreme case of it. It is mainly a concern in people with like severe diabetes, because with diabetes, it kind of inhibits your ability to like detect sensation in your extremities. So for example, if somebody has severe diabetes and has a lack of sensation in their foot, and then they were to get a cut on their foot and that cut got infected, they may not be able to feel that. Again, hopefully that would be caught before this point, but not always. So the reason that Clostridium perfringens is so rare is, again, because it's environmental, and it's, again, very difficult infection to establish because it has to enter via a deep wound. But yeah, why is it called gas gangrene? Again, if you like sort of like gross stuff, the reason it's called gas gangrene, there are many types of gangrene. There's like dry gangrene, which is this. There's wet gangrene, which is like not dry. You could assume like wet, like pus. And then gas gangrene is gas because the drying out of this tissue actually traps any other gases below this tissue. So characteristic, if gas gangrene is suspected for the infection, one of the things you could do is you could palpate or touch the wound. And if you heard like crackling, that would be gas trying to escape between the tissues. So that's where you get the gas gangrene. Two other types of organisms that form spores and are of concern that are normally prevalent in the environment, but can cause wounds. Again, these would really only enter through puncture wounds. I'm sure that several of you can kind of guess what I'm going to talk about next if I'm talking about puncture wounds. Tetanus is also a Clostridium, so also a bacteria that forms endospores. So you can see the spores here. Clostridium tetanus causes like uncontrollable muscle contractions. And so this is sort of like a historical painting of a person with early stages of tetanus, the muscle contractions causing this sort of contracted posture, as well as, I don't know if you can see too much of the face, but one of the first symptoms of tetanus infection is lockjaw, where you get this kind of like tense face. Tetanus shot is one of the most kind of like common shots that you would get. So it is included in the DTaP or the Tdap, several vaccines, and it does need a booster about every 10 years. And a lot of times they'll give you a booster even if it hasn't been 10 years. So for example, if you're going to go in, it's like you have a deep puncture wound that you like maybe got in the environment. A lot of times they give tetanus shots prior to a surgery or to like giving birth, just to make sure, because you're exposing your body to kind of areas where, or areas of your body that are not normally exposed to oxygen. And I know we're running out of time, so I just want to mention also Clostridium botulinum is also an organism that causes botulism, which is the exact opposite of tetanus. Instead of muscle contractions, this causes muscle relaxation. And actually we use the toxin from this for Botox, relax those muscles so they don't wrinkle. I don't know. But I have a picture of honey here, because if you've ever heard that you shouldn't give like babies or children under a certain age honey, it's because honey is normally is an antibiotic, actually, because it is so hypertonic that the majority of bacteria, if they're exposed to honey, the hypertonic solution that honey is will actually dry out any bacteria, killing them. The only bacteria that will be able to survive in the presence of honey would be those that can form endospores, which would be like tetanus or botulism. And so that's one of the reasons they say, like, don't give that to children. Their immune systems haven't formed enough to where they could combat that disease, actually. So yes, I ran out of time, but I just wanted to, oh my gosh, Linda, I'm so sorry about your friend that had the leg amputated. Yeah, that's awful. Yeah, diabetes is where you would normally see gas gangrene. Yeah, all right. And yes, Ren, exactly. That's why pool rescues are so timely. Yeah, I'm glad to hear that everything is okay with the fire alarm. I hope everybody is doing okay. Let me know if anything, it is recorded, so yeah. All right, well, we are out of time. I'm really sad about that. That was really fun. Have a wonderful day. I can stick around for a couple of minutes if anybody has questions or anything like that. I'm glad it was a drill. Perfect timing. But yeah, thank you all for coming today, for making my day. You were all so fun and wonderful. Have a good day.
Thank you.
Thank you. Have a good day. Bye.
I have a question about the lecture.
Yeah.
How would you kill those cells if they can form those spores and survive in like different solutions, like hypertonic solutions?
They're very, very, very difficult to kill. So for both Clostridium and botulinum, well, actually any spore-forming bacterium, there are very specific, well, one, actually, if you have, they actually, I know this is like difficult for me to like say, I apologize. They actually characterize antiseptics and disinfectants, mainly disinfectants, by whether they can kill spores or not. So if you hear something is a high level disinfectant, that actually implies that it's strong enough that it can kill endospores. So there are very few disinfectants that can do that. They would be called like sporicides or sporicidal. But really, like in a lab setting, the only way that you can usually kill spores is going to be using like high heat. So if you've ever heard of like a autoclave, which is like high heat, high pressure, it basically just like without fire heats things up to like over 2000 degrees. Or like flaming something, so using fire. Those are the two kind of physical ways to remove spores. Chemicals would be sporicides. I think like formaldehyde is actually a sporicide. So one of the challenges with those, though, is that those are so strong. Obviously, like if a wound has a spore in it, you can't apply formaldehyde or fire to the wound. Cauterizing used to actually be a strategy for like battlefield wounds and stuff like that. So where you would burn the wound to clot it, but also to kill any spores that had got into it. But now, like tetanus used to be a huge concern, but it's less so, mainly due to the vaccine. So it's really difficult to treat those with antibiotics. And actually with tetanus specifically, I think like I'm not sure about botulism, but I know with tetanus, if you get infected with the bacteria, the muscle contraction is actually not directly related to the presence of the bacteria. It's due to a toxin that the bacteria produces. So you could get a tetanus infection and then you would have to take like an antitoxin. But if the toxin actually binds to your body, it binds irreversibly. So those muscle contractions like won't stop. Like the only treatment is muscle relaxers. So antibiotics won't even cure it. So it's actually really scary, but that's why why it's so important to like get the vaccine, is because the vaccine actually just like prevents you from getting it in the first place, because there's no treatment for it. There's only a preventative for it.
So when they bind, that's irreversible. So the only way you can treat it by then is just using muscle relaxers, because it's gonna keep on contracting.
Yeah. Mm-hmm.
Is that also why some people have to have their limbs like amputated?
So limbs would have to be amputated. So with tetanus, you don't really get limb amputation.
It would just
That is
contract,
more with
right?
like gangrene. But yeah, I mean, if with that slide that showed gangrene, the reason you would have to get that limb amputated is because you can't replenish that tissue. Like that tissue is dead. So it can't be replenished. Even if you were to supply oxygen to it, it couldn't basically be brought back to life. So in that case, like your body is like kind of like wasting energy on trying to like supply nutrients to what remains there, like maybe the bones or any like connective tissue. So at that point, it's just like an energetic waste for your body, as well as that it could be a portal for like secondary infections. And so in that case, like usually amputation is like the safest route. Does that make
Okay,
sense?
thank you. Yeah, that makes
Yeah.
perfect sense.
Good questions. Right,
Um,
have a wonderful day.
I had a quick question from the
Yeah.
review.
Yeah.
I got this problem wrong and I wrote it down, and so I wanted to ask about it. It was splitting water with energy from light, and the question was harnessing light and splitting water occurs in both photosystem 1 and 2. I put true because I looked at my notes and it says that photosystem 2 and photosystem 1 are light dependent, and then also that they split water. Why is it wrong?
So I'm actually going to help you with that. I'm going to share my screen because it's actually only the splitting of water actually only occurs in photosystem 2. So if you look here, you see the light absorb. So yes, both photosystem 2 and photosystem 1, both of those absorb light, but it's actually only photosystem 2 that splits the water. And then those electrons enter the electron transport chain or photosystem 1. Does that make sense? So with photosystem 2, those electrons used from the splitting of water enter the electron transport chain. But then they actually subsequently, well, I guess, yeah, subsequently enter photosystem 1, where they are attached to NADPH, which is actually one of the reasons we found out that like photosystem 2 occurs before photosystem 1, because photosystem 1 is kind of like dependent on photosystem 2 for that splitting of water.
Gotcha. Okay, so it's wrong because the splitting of water doesn't occur in photosystem 1, but
Exactly.
the light thing reaction happens with both.
Yes, exactly.
Okay, got it. Thank you so much.
Yeah, no problem. Have a wonderful day.
You too.